Metabolic robustness, the ability of a metabolic system to buffer changes in its environment, is not always a welcome feature for microbiologists. It interferes with metabolic engineering or prevents that antibiotics kill bacteria. Therefore, it is important to understand the mechanisms that enable metabolic robustness. A massively parallel CRISPRi screen demonstrated that E. coli metabolism is very robust against knockdowns of enzymes, and multi-omics data revealed the mechanisms behind it. In the future, the researchers want to apply this knowledge to build better models of metabolism, which enable rational design of industrial microbes. In their natural habitat, bacteria like E. coli are confronted with constant changes in the composition of nutrients. But under laboratory conditions, they can also be real specialists, and grow for instance on a single carbon source like glucose. To do so, their metabolic network must synthesize all cellular building blocks from scratch. This task requires that hundreds of enzyme-catalyzed reaction in the metabolic network work at the right pace, and that no reaction accidentally falls below a critical threshold. Otherwise, a single bottleneck in the network may have widespread consequences and eventually stop cellular growth. To understand how E. coli accomplishes this task, researchers led by Dr. Hannes from the Max Planck Institute for Terrestrial Microbiology applied the CRISPR interference (CRISPRi) technology. By inducing knockdowns of each protein in the metabolic network of E. coli, they created a CRISPRi library with 7,177 strains. Deep sequencing of the library during a pooled competition assay allowed the researchers to track fitness of each CRISPRi strain for 14 hours. The results of this massively parallel CRISPR screen were somewhat surprising. While knockdowns of only 7 genes, key points in the metabolic network, like biosynthesis of deoxynucleotides for DNA synthesis, caused immediate and strong fitness defects, hundreds of other knockdowns had little effects. As Dr. Hannes Link explains, our results demonstrated that E. coli cells accomplish a very high metabolic robustness. In general, robustness enables living organisms to survive despite external and internal disturbances, and there are different mechanisms that mediate it, such as feedback mechanisms or redundancy. In this context, organisms are always in a trade-off situation. Either they express high enzyme concentrations, which is costly, or they express low enzyme concentrations which can limit the metabolic capacity. For us researchers, robustness is not always a welcome feature in bacteria. For example, in the course of biotechnological applications, if we want to engineer metabolism to overproduce chemicals with bacteria. Therefore, it is important to understand how E. coli accomplishes this task. To answer this question, the team measured the proteome and metabolome of 30 CRISPRi strains. In some strains, the proteome responses revealed mechanisms that actively buffered the CRISPRi knockdowns. For example, knockdown of homocysteine transmethylase, MEAT, in the methionine pathway caused a compensatory upregulation of all other enzymes in the methionine pathway. In other words, E. coli cells sensed that the knockdown caused a bottleneck in methionine biosynthesis and then mounted a very precise and local response around the methionine pathway. The other 30 CRISPRi strains revealed similar buffering mechanisms that were surprisingly specific, but whether all metabolic pathways are equipped with such precise and localized buffering mechanisms remains open. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.